Hello, everyone. Welcome to today's webinar. I'm Winton, the product owner of ProteoformX. Okay. In this session, I will give you a complete overview of ProteoformX, our integrated platform for proteoform analysis using intact top-down or middle-down and bottom-up mass spectrometry. We will cover core concepts, the key functions, and the supported biologic modalities and advanced features to help you achieve molecular precision in proteomics and biopharma development. I will also show how ProteoformX integrates these workflows into a single platform and followed by a demo walkthrough. Okay. Traditionally, we talk about proteins, but in reality, proteins exist as multiple proteoforms. So that arise from genetic variation, the alternative splicing, and post-translational modifications, and other variations. Importantly, proteoforms, not proteins, drive a biological function. In 2022, a large-scale study have shown tens of thousands of proteoforms from a limited number of genes. So this highlights a key gap. Protein-level analysis is not enough for precision biology or biopharma characterization. So ProteoformX bridges proteoform analysis across both research and biopharmaceutical applications. It integrates workflow for fundamental proteomics research and biopharmaceutical product characterization. By integrating multiple mass spectrometry strategies, it solves complex challenges across research and development. Since ASMS in last year, we released the ProteoformX 1.0 version, which focused on the intact mass and top-down analysis. This week, we are excited to introduce the version 1.5 with expanded capabilities. So today, I will walk you through the overview of the ProteoformX and one of the workflows, the new peak detection. Okay, let's look at the four core functions in ProteoformX. First is intact mass. The intact mass analysis answers this question, what exists? So it provides a detectable proteoform at whole molecular level, enabling rapid assessment of sample heterogeneity. And the top-down search answers the question, what are they? It provides a global view of individual proteoforms with bottom-up data assisting top-down analysis. And the new peak detection. So this function detects the emerging peaks and monitors biopharma metabolites, trends, and product-related changes or impurities. The peptide mapping, we both know peptide mapping as use the enzymatic digestion to characterize protein sequences, post-translational modifications, and sequence variants at the peptide level. And we use the protoform manager to define the expected protoforms to validate. Okay. Before diving into the detailed functions or features, I would like to briefly introduce the core algorithms behind ProteoformX. Across all workflows, intact mass, top-down, and bottom-up based workflows. And for both identification and quantification, ProteoformX is built on a feature-based approach. So this process begins with feature detection, where signal are extracted from the LC mass data, and the features with the same mass across a different charge states are then grouped into what we call the feature groups. And for protoform analysis, these feature groups represent candidate protoforms. And by associating mass spectrum, that identifications can be assigned to individual features or feature groups. Finally, relative quantification is performed based on the peak area of the detected features, enabling comparison across samples. So that is a algorithm basic. And for each function, I would like to give you more detailed features and something unique in ProteoformX. And let's see what advanced features makes ProteoformX is so helpful. The first workflow is intact mass. The key advanced features include we can provide a high precision mass deconvolution algorithm and give you comprehensive sample annotation with modifications. And also for the glycosylation, we can give a profiling at the proteoform level. And I highlighted something new in this version that we also provide a function that we called manual annotation editing. That for experts that they can make sure the annotation whether is they expected. And also, we support the selective digestion in the protoform manager to generate like a subunit. And we also input. We also support. You can input the FASTA sequence to detect protoforms directly. So these improvements greatly boost accuracy and flexibility. So here is a result overview for the intact mass, and I would like to highlight the unique features of intact mass that we have that in this version we provide this manual annotation editing function. So this function can help user to decide the annotation of the deconvoluted mass peak. So this is particularly important for complex moleculars, such as multiple payload ADCs, where a single observed mass may correspond to multiple candidate proteoforms. So by default, the software assigns the annotation with the smallest mass error between the theoretical and the measured values. However, this may not always reflect the expected or biologically relevant assignment. So with this new function, user can manually select alternative candidates based on their knowledge, or even you can directly input a custom assignment. So, for example, here is an ADC with a payload, and sometime the payload the drug may cleavage from the payload. So you only have the linker. So for this annotation, then you can use this manually annotation function to select what is your expected protoform. And in additional, an auxiliary line is provided to help calculate and visualize the mass differences between adjacent peaks that facilitating the identification of patterns such as payload increment and modification series. And ProteoformX uses a bottom-up assisted top-down or middle-down approach. So here is some highlights include we support for diverse fragmentation method and we give you a hierarchical tracing from the feature or feature group to the protoform and to proteins. And also open search for unbiased PTM and variance discovery. So in the summary page, we can give you like some the distribution of the protoform types, such as if you get the protoform with a full length, or this is some N-term segment or C-term segment or some middle segments. And here is an example for hierarchical information with feature-based identification algorithm. Flexible user interaction that enables the linkage between different views, like from the LC-mass view, the spectrum view, the PRSM, and also we integrate the top-down and bottom-up to show the coverage for the protoform. And also we mapped all of the protoform to its related proteins. Well, nowadays, for next generation therapeutics, there are many novel biological molecular type, increasing complexity that requires more flexible and accurate analysis tools. For example, the peptide-based drugs, including unnatural amino acid, and some oligonucleotide nucleotide conjugates, and also for maps, both symmetric and asymmetric chains, or some antibody drug conjugates, antibody peptide conjugates, or even antibody oligonucleotide conjugates, such as antibody RNA conjugates. So ProteoformX support those next generation biological moleculars analysis. So for the novel molecular characterization, we should or we need to ask to answer this question: what's the new or unexpected? That includes like a new peak detection or open search. To address this question, we provided two new workflow that named one is new peak detection, and then one is peptide mapping. So in those two workflow, not just for identification, I'll also give you some quantitative insight so that we can compare relative abundance across samples. So those two workflows give us some more detailed information, like for the new peak detection, we support the peptide, including a natural amino acid or a natural nucleotide. In this function, we also support not only for the linear peptide, but also for branched cyclic peptide analysis. Then we quantified the feature between the different samples and show the variation trend. So we can get some biopharmaceutical metabolites trend through the time series. And we also support the negative acquisition mode, and we can get more information such as relative abundance and the retention time between the target molecule and its product molecule. In this function, we have some more manually validation function that such as manual feature editing, or we can call it a manual feature extraction, or for the result release. And the peptide mapping, I think people are very familiar with this function. In this function, ProteoformX provide not only for the normal unexpected modification or sequence variants, and also we integrate the open PTM in this function. And we use some representative peptide to show the variation between the different samples and report some atypical result in batch control that will help the QC or QA analysis. And then in this function, we provide some different kind of PTM profiling function that we also can help you to build your customized report. So considering the time issue, so I will just pick one of those workflow to give you more detail. So let's go next to the new peak detection. For the peptide therapeutics represent a class of biopharmaceuticals of significant importance in modern medicines. However, traditional software for mass spectrometry data analysis are limited to the canonical amino acid and linear peptides. So the new peak detection, this function is essential for the biopharmaceutical development. It identifies new peaks, tracks the metabolites variation trends, and flags the product-related impurities or changes over time. Here, I would like to give you an example. We both know the semaglutide or tirzepatide, those two widely used GLP-1 receptor agonist for obesity and diabetes treatment have gained significant attention in recent years. And their representative example of peptide-based biopharmaceuticals containing a natural amino acid. And also, it has like the 18 carbon fatty acid side chain in the semaglutide, and the 20 carbon fatty diacid group linked to, via gamma group with 2 AEA at lysine at the 20th lysine in tirzepatide. So those kind of tailored modifications prolongs in vivo half-lives and optimize therapeutics efficiency. And in those kind of molecules, an alanine at position 2 in the semaglutide, and in the position 2 and 13 in the tirzepatide were replaced by 2-amino acyl butyric acid. So before we start, so we needed to define those amino acyl butyric acid as a natural amino acid in the ProteoformX, and also those modifications. So to simulate in vivo metabolic processes, we use the semaglutide and the tirzepatide that processed that digested by chymotrypsin that about 40, 30, 60, and 90 minutes. So the raw data were analyzed using our new peak detection workflow, so that we can get those results. Here I show you the feature table. So in the feature table, we give you very in detail about the target molecular and its products related moleculars. So in the panel A, we can see this feature table, which lists all detected feature vectors after alignment. And for this, each row represents a feature vector, and according the information level, all of those columns were defined as master columns and the sample level columns. The master columns shows overview of the feature vectors, and the sample columns, sample level columns shows the feature information from a specific sample. So in the master columns, that including the such as sequence, mass error, score, and the abundance this information, and also we provide some relative information between the target molecular and its products. So these features are grouped into compound. Then we can get the compound table here. For the compound, that means we can get the same mass from those feature across different charge states. So in this view, you can quickly filter and sort all of those candidates or features based on the score or the areas, which means abundance or mass accuracy or modifications. The panel B shows a variation trend of the selected feature across the time point or between the different samples. Here we clearly see the decreasing trend, which may indicate metabolic degradation. So this enables time course analysis and metabolite tracking. And the panel C here displays the XIC, the peak view, which displays the isotopic pattern of the selected feature vector, and the LC-mass view across different samples. So you can visually confirm the retention time and the MOVZ consistency of the features. So this helps validate that the signal is real and consistently detected rather than noise. So visual validation across samples is critical for confidence. Then the panel D shows the mass1 and the mass2 spectrum. The mass2 spectrum was annotated for the sequence confirmation. And for the fragment ion are matched to this sequence that allow us to confirm the peptide identification result and also for the modifications. And here we also have the mass arrow map and the ion matched table here to provide confidence in identifications. In these functions, we developed syntax to present the linear peptide and also the branched peptide or cyclic peptide. That make more easy to input sequences in the FASTA format. So this sequence can contains the unnatural amino acids or unnatural nucleotides. And the ProteoformX that enable you can directly to use this linear syntax to represent such complex moleculars. For the branched peptide, we use the square bracket to show that this part is connected to the this side chain of the this main chain. And for the cyclic peptide, if it cyclized by head-to-tail peptide bond, and we just use this bracket to show okay, this PEPTIDE was cyclized by this P and E to generate the peptide bond. And the cyclic peptide, if it connected by disulfide bond between the cysteines, and we also use this bracket, but here we added the number to present the cyclic peptide connector between which cysteines. So here is an example like CYIN CPLG. So those two cysteines connect by disulfide bond. So you just need to write down the number 1 into this bracket, and for another cysteine, and you write down the same bracket with and for molecules with complexity sequence and structures, here we also support the cross-linked cyclic peptides. And we allowed the branch attached. Here the syntax is we use a lowercase, which defined that in UAA configuration, that means the amino acid. We use this configuration to present the chemical group as a linker. We support the branches on a main chain or in the ring. The first example is here we have a peptide. In this example, there is the actual linkage between those two cysteines. But the mass shift comes from minus to hydrogen plus methylene group. So there is only a carbon remains. So we define this carbon as a A. So we use this linear syntax, the AYC bracket 1A, PPTIDE and the bracket 1A to present those cysteine have a cross-linked linkage that to form a cyclic peptide. So we only keep the residue of the chemical group to calculate the mass. Because there are so many molecules with such complex sequence and structures. So we also provide a dedicated tool that we named mass validation in our software. In this function that helps us to verify the calculated sequence mass based on the input sequence, and we will generate a comprehensive peptide list, including all subsequences produced by unspecific digestion. So this sequence mass list will displace prefix subsequences generates through C-terminals cleavage, and all the subsequences list can be exported as a CSV file. So this tool will help user validate the sequence, whether the input, the sequence is correct. And also we support this branched peptide in this cyclic peptide. So here is another example. The last example is for the peptide oligonucleotide nucleotide conjugates. So for the nucleotide, for the POC, that we define this as a three subunit of a nucleotide. One is the backbone linker, and the ribose and the base. So we use a triplet method to present this sequence. For example, the 5' P uppercase A, the capital A, and the lowercase r, and for another P and UR or PGR, and we use a 3' prime as a end of the oligonucleotide part. And here it conjugated with a peptide, and we just use a normal syntax to present the peptide with the capital letter for the amino acid. Well, in the new peak detection, we have this unique feature that we call the manual editing. Because the automated feature detection may miss certain features due to factors such as low concentration of the degradation products, the peaks ship interference, or some matrix effects. So these issues can promise the comprehensive screening of the degradation products and reduce the accuracy for the of the quality control result. So we use this manual editing of feature detection function. So this function is designed to address these challenges by allowing users to manually and accurately select and define those incorrect detected or previously unidentified features in this target chromatogram and mass spectrum. So after the software's automatic detection, we can use this function. As an example here, we can see this variation trend of intact cyclic peptide that appeared unexpected at the 30 minutes time point. So the LCMS view show us the RT range here for this feature in the 30 minutes time point may have some issue, or it may be interrupt. So we just click this manual editing button that we can access this function here. So in the this view, and we can first we can based on this peak view window that we can right click to select a peak as or set it as a M0 peak. Then define the charge state of this this feature and the number of peaks. Then those selected peaks will be added to the isotope peaks list in this table. After manually redefining the the retention time range, and we apply it to the specific sample, or we can apply to all sample. Then we click the preview button that we can see the variation trend. The variation trend changed, and the cyclic peptide trend for those cyclic peptide trend returned to the expected patterns. So this is the general introduction of the ProteoformX and the new peak detection. I will go walk through for the software and give you more detail about those functions in the ProteoformX. So ProteoformX enables, as a conclusion, ProteoformX enables comprehensive, flexible, and precise proteoform analysis. So we provide the multiple level analysis and give you a flexibility for complex moleculars, and also the biopharma ready workflows. Okay. Here is our software. We have the projector, protoform manager, configurations, tools, and help those tab. Um, Before we start, we can have a glance of the protoform manager. In the protoform manager, we defined a protoform what we expected. So as an example, for the antibody, the next antibody, here we can here we start create antibody form, and you just double click and rename it. Okay. Um, Then we can directly import the FASTA sequence format file, and also we can use the copy paste to define the heavy chain and the light chain. Before next, do not forget to name your antibody form. So I would like to define a glycoform here. So you just name it, and in the category, you can select the antibody, the map, or recombinant proteins, or some other kind of antibody or protein form. So after click OK, then we have some linkages or some site-specific modification, cleavage site, and a glycan setting here. For the linkages that we can upload the template. Currently, we support the human IgG uh, template. So you can directly select like IgG1. We will generate the densified bond automatically. Okay. Here in the overview that you can see the densified bond between the different systems we can show here. And for the site-specific modification that you can add what you expected into here. You can also right-click for the specific amino acid. Then select the modification and click OK. And also for the cleavage site or the glycans, the process is similar. For the cleavage site, you can just right-click and select this site between 2G. So we will generate a subunit for this heavy chain. And here we find a glycosylated site here, and you can click here and to define some common glycans like G0, G0F, and G1F or G2F. So after we define this protoform, and in the overview, you can find the both the site-specific modification, the linkage, and also we give you overview of those glycoform. That in summary, we give you the mass for each chain and the whole intact mass. And also for the cleavage site, the site-specific modification and glycan or linkages. And also you can define a antibody group in this software. For example, here we have an asymmetric chains antibody. So you can see we defined those two different LC and HC. Then we go. Okay. Then we continue for the configurations here. And for the unnatural amino acid here, we just click this icon. Then we go to the UAA list, and you can create a new that we name the UAA, and you just input the chemical formula of the UAA residue. You will directly got the monomass, and we just needed to give the UAA abbreviation to present this UAA. For example, I give this a G as a abbreviation. Then I click add. So you will update your UAA list here. And also for the unnatural nucleotide is similar with the unnatural amino acid configuration. For the glycan, we built in some unlinked and all-linked glycosylation. So if you have some other glycans that you want to add in, you just use this new button to access the glycan editor. So we use this draw board that you can create a new glycan. Just click this triangle, square, or a circle here. And also in the tool, I think we're both familiar with the calculator, the work monitor, and then in the ProteoformX, we have the audit log that follow the 21 CRF part 11 that you can got the log file for each process in very detail. Okay, here is an example for the intact mass that in this data set we have the whole antibody, the reduced antibody, and the subunit. So in here, you can select the annotation component, both like glycans, disulfide bond, or some modifications involved in this analysis. And I will use an ADC antibody as an example to the manual editing annotations here. For this ADC, as in our knowledge that we can know there should be DAR 8, the DAR 7 group, the DAR 6 group, the DAR 5 group. But here we can see there is a weird annotation here for this group. And okay, you just click this tag here, and you can find the other candidate annotations, like for the MCCDM1, like for this 4 payload here, and we also have the two G0F glycan attached here. Okay. Here we just modified for those annotation. And for each annotation text, you can use your mouse to drag this tag and to put it in a perfect somewhere. And also you can select or check this accession so that can be the optional that you can export the image that to get your mass deconvolution result for this data. And for the natural amino acid, first I just show you some result about the semaglutide. Here we can get the summary. In the summary page, we will give you statistic of variation trend, the distribution of feature detection, and some other parameter informations here. In the feature table, you can use a feature vector to show to filter all of those the feature vectors. And here we can to check for the each feature vector with a mass spectrum annotation, and also for the in XIC, the peak view, the LC mass view, and here you can select which sample you want to display. And here I would like to show again for the cyclic peptide this example for this manually editing function here. So for this feature, you can see the retention time just interrupt. And here you can switch to the 3D model that if we check the unclassic view, then you can got this 3D view here. For this 3D view, you can use your mouse directly to drag this retention time to change it to the retention time range, and more easier way to define this feature area or the peak the MOZ and the isotopic pattern. You can just select one of those samples and apply to all samples. Here you can get the preview if you check this is correct or what is your expected. You can just click this save or you can click this save as. Then we will modify these features. Also, we have some mass spectrum annotation for the cyclic and others complex sequence or peptide here. And one more thing is here we in our mass spectral annotation we can define a confirmation ion here. Like you can see, there is a test here that is defined by users. That means we have some fragmentation ion that's related with those features or those compound that helped you to confirm your compound is really existed in your samples. I think that's all from me. Okay. Thank you.